Isolation of Francisella tularensis outer membrane vesicles and their associated outer membrane proteins starts with osmotic lysis of bacterial cells. Logarithmic phase bacteria are sequentially treated with hypertonic sucrose, EDTA, and lysozyme solutions before being slowly diluted into deionized water. The resulting bacterial lysates are subjected to low-speed centrifugation, which removes cellular debris and unbroken cells, followed by high-speed centrifugation to pellet membrane vesicles. Next, total membrane vesicles are layered onto sucrose density gradients in order to separate inner membrane, or IM, and outer membrane, or OM, vesicles. OM vesicles can be separated from IM vesicles based on differences in buoyant densities, believed to be predicated largely on the presence of lipopolysaccharide, or LPS, in the OM. Immunoblotting of sequential fractions from sucrose density gradients shows spatial separation of inner membrane proteins from outer membrane proteins.
Hi, I'm Dr. Michael Norgard, and this is Dr. Jason Huntley from the Department of Microbiology at UT Southwestern Medical Center at Dallas. Today, Dr. Huntley will show you a procedure for extracting, enriching, and isolating Francisella tularensis outer membranes and their associated membrane proteins.
We use this procedure in our laboratory to study the outer membrane localized and potentially surface-exposed molecules for this intracellular bacterial pathogen. We have a busy day, so let's get started.
To begin this procedure, prepare and grow 2 500 milliliter cultures of F. tularensis bacteria as described in the accompanying written protocol. Retrieve the bacterial cultures from the shaking incubator and remove a 1 milliliter aliquot from each culture for OD measurement. F. tularensis cells in early logarithmic phase of growth, correlating with an OD 600 between 0.2 and 0.4, give optimal membrane extraction and isolation results. When the cultures reach the desired OD 600, use 4 250 milliliter centrifuge bottles to centrifuge the cultures at 7500 Gs for 30 minutes at 15 degrees Celsius in order to collect the cells. After centrifugation, carefully remove the media supernatant from each centrifuge bottle and firmly tap the bottles on absorbent material to remove excess growth medium. Within 10 minutes of completing the centrifugation, suspend each bacterial pellet in 8.75 milliliters of 0.75 molar sucrose in 5 millimolar tris pH 7.5. After suspension of each pellet, transfer the solution to a sterile 250 milliliter flask holding a small stir bar. Repeat for all 4 bacterial pellets with a final bacterial pellet suspension totaling 35 milliliters. Thoroughly mix the cell suspension on a stir plate, but not too fast in order to avoid frothing or bubbles. While mixing, slowly add 70 milliliters of 10 millimolar EDTA in tris to the solution over the course of 10 minutes. Hold the tip of the pipette below the cell suspension level to avoid elevated local concentrations of EDTA. After adding the EDTA, incubate the solution for 30 minutes at room temperature. Then, while still mixing, slowly add 11 milliliters of a 2 mg per milliliter lysozyme solution to a final concentration of 200 micrograms per milliliter. Continue to mix the cell suspension during the lysozyme solution addition. As before, hold the tip below the cell suspension level. Incubate the cells with the lysozyme for 30 minutes at room temperature. During this incubation, prepare a sterile 1-liter flask with a small stir bar and 530 milliliters of room temperature cell-grow molecular grade deionized water. When the cells have finished incubating, osmotically lyse the cell suspension. Using a 25 milliliter pipette resting on the bottom of the flask, adjacent to the stir bar, slowly dilute the cells into the water in the 1-liter flask over the course of 10 to 15 minutes with gentle mixing. This step is challenging and is critical for overall success of the experiment. Stirring should be fast enough to thoroughly disperse the cell suspension, but not too fast leading to frothing or bubble formation. As needed, slowly increase the stir bar speed to ensure proper mixing. Carefully monitor the dilution process and adjust the flow rate or rotate the flask as necessary to disperse local accumulations of cells, cell clumps, and cell wisps. Once all the cells have been added, incubate the mix for 30 minutes at room temperature. At the end of the incubation, transfer 40 milliliter aliquots of the osmotic lysis solution into 50 milliliter conical tubes and centrifuge at 7500 Gs for 30 minutes at 10 degrees Celsius to remove intact cells and debris. Following centrifugation, carefully remove 27 to 30 milliliters of supernatant from each conical tube and pool the supernatants in a sterile 1-liter flask. Transfer 25 milliliters of pooled supernatant into each of 16 ultracentrifuge tubes and centrifuge at 200,000 Gs for 2 hours at 4 degrees Celsius. 10 minutes before centrifugation is complete, prepare modified membrane resuspension buffer. Combine 8 milliliters of membrane suspension buffer with 1 tablet of EDTA-free protease inhibitor cocktail and 375 units of benzonase in a sterile tube and mix gently. Following centrifugation of the lysed cells, proceed to recover the membrane pellet. When ultracentrifugation of the lysed cells is complete, carefully pour off the supernatants, then invert the tubes on absorbent material and firmly tap them to remove excess supernatant. Mark the location of each membrane pellet with a marker to aid in visualization. Gently resuspend 8 membrane pellets with 600 microliters each of modified membrane resuspension buffer. It is difficult to resuspend the membrane pellets, so continue gently passing the modified membrane resuspension buffer over the pellet until it peels away from the wall of the tube. This is another step which is challenging and critical for overall success of the experiment. If necessary, use the micropipette tip to scrape the membrane pellet off the tube wall. When pipetting, avoid frothing or bubble formation. Excess shear forces will lead to mixed micelle formation, which precludes membrane separation. Transfer each membrane resuspension to the next set of 8 tubes, gently resuspend the remaining pellets, and pool all suspensions in a sterile tube. After removing the membrane pellets from all 16 tubes, wash every 4 tubes with 600 microliters of modified membrane resuspension buffer. Focus the wash around the marked area of each tube and combine the wash with the resuspended membranes. Repeat the wash for the remaining tubes. Incubate the membrane resuspension with gentle rocking for 30 minutes at room temperature to degrade DNA and aid in reduction of flocculent material. Next, remove a small aliquot of the membrane suspension and perform protein quantitation to determine total membrane yield. Prepare 1:1 and 1:3 dilutions of membrane suspension in 2.5% SDS. Heat the undiluted 1:1 and 1:3 membrane suspensions at 90 degrees Celsius for 10 minutes. SDS and heat are required to release OMPs from the extracted membranes. Use the Biorad DC protein assay to quantitate the membrane resuspension protein concentration. Total protein yield is generally between 1 and 1.6 milligrams per milliliter. Once the concentration of protein in the membrane preparation is determined, the samples can be separated on a sucrose gradient. Start by preparing a series of sucrose solutions in EDTA. To prepare the linear sucrose gradients, use a bulb pipetter to layer 1.8 milliliters of each sucrose solution into 14 by 95 mm ultra-clear ultracentrifuge tubes in the following order: 55%, 50%, 45%, 40%, 35%, and 30%. Next, layer 1.5 milligrams of membrane resuspension on top of each gradient. Do not load more than 1.7 milliliters of membrane resuspension per gradient. Individually weigh each tube and carefully adjust the final weight by adding or removing membrane resuspension so that all tubes weigh the same. Transfer the sucrose gradients into an SW40Ti swinging bucket rotor and centrifuge at 256,000 Gs for a minimum of 17 hours at 4 degrees Celsius. After 17 hours of centrifugation, stop the centrifuge run and carefully remove the sucrose gradients from the rotor. Next, collect the fractions. Carefully clean the bottom of the sucrose gradient tube with 70% ethanol, then puncture the bottom of the tube with a 21-gauge needle and collect sequential 500 microliter fractions from the gradient into sterile microfuge tubes. The gradient should be allowed to drip by gravity flow so as not to disturb the sucrose gradient. However, if the dripping ceases from the bottom of the gradient tube, a small amount of pressure may be applied to the top of the tube using one's finger. Finally, determine the refractive index of each sucrose gradient fraction using a refractometer. Correlate the refractive index with a specific density in grams per milliliter. Assess protein localization of representative sucrose gradient fractions by SDS page separation and immunoblotting. Here is an immunoblot of fractions from the sucrose gradient used to separate the membranes extracted from lysed F. tularensis live vaccine strain. F. tularensis outer membrane proteins, or OMPs, localize between densities of 1.17 and 1.2 grams per milliliter. By comparison, inner membrane proteins, or IMPs, localize between densities of 1.13 and 1.14 grams per milliliter. Similar results were obtained for the F. tularensis Shu S4 strain.
I've just demonstrated how to extract and isolate Francisella tularensis outer membrane vesicles through osmotic lysis and sucrose density gradient ultracentrifugation. When performing this procedure, it's important to remember to use bacterial cells in the early logarithmic phase of growth, to slowly but consistently dilute those bacterial cells into deionized water during the osmotic lysis step, and to take care when resuspending the ultracentrifuge membrane pellets. This concludes our membrane separation procedure. Thanks for watching, and good luck with your future experiments.